So this video is a continuation of our biological molecules lecture. So this is part 2. In the first part of the biological molecules lecture, we talked about monomers and polymers. We talked about carbohydrates, and we talked about lipids. So if you haven't watched that one, I recommend watching the first part first and then coming back to watch this part. So we're going to continue on here. We're going to talk about the proteins, and we're going to talk about the nucleic acids. So let's go ahead and start with proteins. Remember, again, monomers are the building blocks, mono meaning single, or one, so it's the single pieces of our biological molecules that are building up to our polymers, which are many parts. So our monomers of proteins are the amino acids. And I'm not going to go over all of the different structures of the amino acids, but I am going to draw a basic amino acid here just so that we can take a look at it. You don't have to have this memorized or anything. It's just so that we can talk about it, so you can understand what it is that we're seeing here and how it's going to be interacting. All right, so when we are talking about our proteins, the function of proteins, there are many, many functions. So everything from metabolism, because they make up enzymes, which we'll talk about separately, support, because I can write some of these down. Let's see, metabolism. Woo. All right, metabolism. Support, because they make up the structure of our hair and our nails, the outer part of some of our rough parts of skin. Transport, because we use them to move things throughout our cells, but then also in and out of the cells. Also defense, because we use them to make antibodies, which are going to be part of our immune system. They're going to help us fight off different things. Regulation, because we have hormones that are made from proteins. Let's see, regulation. Motion, for movement, because we have actin and myosin proteins that are in our muscle cells that we'll talk about that are going to attach to each other and move our muscles, within our muscles, so contracting our muscles for movement. So these are some of the different functions of proteins, but really proteins are integral to the body in that they are used in many, many different ways all over the body. So getting back to our monomer of protein, this is our monomer of protein here. This is our general amino acid. And all of our amino acids have a similar structure in that they have this central carbon here. Coming off of this central carbon right here, this is what's called an amino group. It's an amino functional group, and we didn't go over the functional groups, and that's okay, but it has an amino group, which gives it its name. It's an amino acid. And then up here, this is kind of our acid part, our carboxylic acid functional group, or carboxyl group. I'll write that down, carboxyl group. Again, you don't need to know these different parts. This is just so that as we're talking about it, you can understand why things are functioning the way they are. Right here, and then we have a hydrogen here. And then right here, this R actually is representative of the rest of the molecule. So this is where we have differences in the different amino acids. So all of these other parts we already talked about. So this hydrogen over here, this carboxyl group at the top, and then this amino group over here, including this central carbon, are all going to be the same from amino acid to amino acid. Different amino acids, or the differences between the amino acids, are here at the R group. So sometimes we see something as simple as a CH3 here, so just a carbon with 3 hydrogens on it. That would be alanine. Sometimes we have something like a CH2 with C double bonded again to the O, kind of like the carboxyl group up there. That's aspartate. Sometimes we see really long chains similar to our fatty acids, so we see a lot of carbon chains coming down here. If I kept going, that would be lysine. So we have different amino acids, and all of these chains are going to be connected to this central carbon. But these are the different types of R groups, for example. And as you can even see here in some of these examples that I drew, like this one, for example, we can see that it has a negative charge here. So you can imagine that this amino acid would be a polar amino acid, whereas this amino acid, this part of the amino acid, would be non-polar, right? Because it's a bunch of carbons and hydrogens, similar to our fatty acid group. If we look at the basic structure of the amino acid, though, you can see even up here, our amino group, we have a positive nitrogen here, and then we have our negative oxygen here. So it's already a polar molecule in kind of the main part of the molecule that's similar across the amino acids. However, our side chains or side groups, this R group here, can be polar, it can be nonpolar, or it can be a charged group. So it can be negatively charged or it can be positively charged. So this R group here then is what's going to give different characteristics to each amino acid. That's how we name them differently. But it also determines the structure of the protein. So let me get back to the monomer part. So I'm going to just kind of draw this little circle. That's this right here. This circle is going to represent this whole thing up here. Okay. So we have our individual amino acids that we're going to put together. We're going to create what are called peptide bonds between the amino acids here. So if we have this chain of amino acids, these are our monomers, the amino acid, and we put them together into our polymer. And our polymer is just this polypeptide. So polypeptide is our polymer. Our monomer is the amino acids here. So our polypeptide is this string of amino acids that are held together by covalent bonds, by these peptide bonds that are covalent. And we have the attachments, and then what we see is that we have the R groups kind of hanging off of our amino acids, right? So each one, again, has a different type of R group. And these different types of R groups are going to give us our protein shape. And what I mean by that is if it is a polar R group, let's just say this one's a polar R group, then it's going to play well with water. It's going to try to go toward the surface that has water when it is folding. So this polypeptide chain is going to fold and bend on itself, and a polar side chain here is going to go toward water. So if we can then say, let's say this one here is nonpolar, this nonpolar one then is going to probably curve in to try to be by other nonpolar side chains. Just like we saw with our phospholipids, all of those fatty acid chains wanted to stay together. They kind of wanted to sequester themselves. We see the same thing with our nonpolar side chains. And then our charged side chains, since they're charged, they would play well with water. So they would also want to bend and move toward the water. And so if we have a mix, if you can imagine, we have these really long chains of amino acids, all with different R groups here, polar, nonpolar, charged groups. What ends up happening then is these bend. They'll bend on these peptide bonds here, and they'll move and they'll form different shapes. And so we have different levels of protein structure then. Our first level, or our primary structure, our primary protein structure, is this right here that we're talking about. Our primary structure is simply one amino acid attached to another, attached to another, attached to another, making up our polypeptide. So our primary level of protein structure is our polypeptide chain. Okay. Then when we're moving into our secondary structure, our secondary structure is going to be this interaction that I'm talking about, these interactions. So these are going to be the hydrogen bonds that are between the amino acids. These are going to be causing the chain, the polypeptide chain, to then be bending and moving. Okay, so the first interaction is actually going to be with the hydrogens, and basically our hydrogen bonding. So our water bonds that we talked about in the chemistry section. So we're going to have these interactions between these partial positive and partial negative pieces. And the partial positives are going to be attracted to the partial negatives. And what we see in our secondary structures, we can form two different types of structures. Our primary structure is just a simple chain of amino acids. Our secondary structure can be kind of this curve like this, which is called an alpha helix. So this is our alpha helix, a helix meaning this kind of spiral shape, if you can imagine, kind of like the spiral part on a spiral notebook. That's an alpha helice, singular or plural, alpha helix, singular. Or it can fold up into what are called beta pleated sheets. So beta pleated sheets. And a beta pleated sheet is actually where we see our polypeptide chain kind of going back and forth in this direction. So this right here, this is a three-dimensional, like our spiral notebook. This here is more like if we took a string or a piece of yarn and we laid it down on the floor and kind of went back and forth on the floor. So it's like a sheet. If we were able to then like dump a bunch of glue on it and let it dry, our yarn, for example, and then pick it up, it would be like a sheet on that case. So our beta pleated sheets versus our alpha helices, which would be like if we took that same yarn, dipped in glue, and wrapped it around a pencil and then let it dry and then took it off the pencil, for example. So our secondary structures, due to this hydrogen bonding, are partial positive, our partial negatives, making our alpha helices and our beta pleated sheets. Then we can move on to our tertiary structure. Our tertiary structure is actually what I was just discussing. I kind of skipped over the secondary for a moment. Our tertiary structure is due to the interactions of the side chains. So I'll write that down, due to the interactions of the side chains. And this is due to whether they are polar, whether they are nonpolar, or whether they are charged side chains. So then what we see is our alpha helices and our beta pleated sheets are going to interact with each other. So we may have an alpha helice here, and then our amino acid chain, depending on the hydrogen bonding, might turn into a beta pleated sheet for a little bit. And then maybe it'll go back into an alpha helice over here, based on whether there are polar, nonpolar, charged interactions, right? So whether or not the side chains are polar, nonpolar, or charged is going to depend on where they're folding and where they're moving. Then let's say there's another beta pleated sheet, and then say an alpha helice that goes over here. And then let's say that's our entire protein there. We have our folding upon folding here. And so then this would be what our protein would look like at our tertiary level of structure, where we have our alpha helices and our beta pleated sheets, and they're now folding together and with each other based on the interactions of the side chains. So at this point, our tertiary structure looks something like this. Then the last part of our structure is called the quaternary, fourth part of our structure, so quaternary. And our quaternary level of structure is when we have two or more folded polypeptides up to that tertiary structure together. So this would be two or more polypeptides interacting. So we would have our folded polypeptide. Maybe it looks something like this. Let's see if I can try to recreate that a little bit. Our folded polypeptide here. Not bad. And then we add another folded polypeptide. And then let's say that looks something similar. And so now we have two folded polypeptides, and they're going to be interacting in this space here. Maybe they're going to overlap in some way due to, again, these side chains. And then let's say we have another one down here, and it's going to interact with both of them, and they have alpha helices and beta pleated sheets, and so now we have interactions here. And so now this one, this quaternary structure, and let's say our final protein, this at this point it's just called a protein. Our final protein, in this case, consists of 3 polypeptide chains. And so this would be our final protein shape. And our shape is very, very important. So an example maybe of a final protein shape that we can think of is hemoglobin. Hemoglobin is actually 4 polypeptides all together as one single protein. And this protein is called hemoglobin. Hemoglobin is actually the protein that is going to assist in carrying oxygen. It's in our red blood cells. It's going to have an iron group here in the heme group, and then it'll attach to oxygen and carry it throughout the body. So that's one example of a final protein. Another type of protein that's important that we will look specifically at are enzymes. So our enzymes are a major player in our bodies. They're what are going to assist us in going through metabolism, breaking things down, building things up. And then when that happens, we have all of these little enzymes that are going to assist in that process. They're going to grab onto our reactants, they're going to have a reaction, then they're going to create products. When that happens, then we are going to be assisting in this whole process of metabolism. And so it's important that we have these proteins in order to assist in metabolism. If we don't have these proteins, then our bodies will be very, very inefficient and we will not be able to do very much. And then, depending on the type of protein, we can actually stop living because they're that important. We're going to talk about enzymes separately, so we can move on now to our nucleic acids. So our final biological molecule then are our nucleic acids. So our major function of our nucleic acids is these are for storing information. And specifically for storing information about how to make proteins. So the basic unit here, our monomer, is the nucleotide. And our nucleotides are going to be different depending on whether or not we're talking about DNA, which is one of the nucleic acids, or RNA, another nucleic acid. We're going to take a quick look at both of them. Our nucleotide is composed of a sugar, a phosphate, and a base, which is a nitrogenous base. So our structure, I'm going to kind of draw a general structure here. We'll just put the OHs down here, here. Just do this as the base here. And then over here, we're going to put our phosphate group. Again, you don't need to memorize this structure in any way. It's just so we can talk about it, so you can understand what I'm talking about as we're talking about it. So the first part is our sugar, and we mentioned when we talked about carbohydrates our ribose sugar, and that's what we see here. Is this is our ribose. Then over here, on the other side here, this is our phosphate group. So phosphate group is another functional group that interacts with other things. So you can see that this is actually charged. We have oxygens there, which are negative. There's negative oxygen here, so that interacts and will form bonds. And then over here, this is our nitrogenous base. And our nitrogenous base is what's going to change depending on which nucleotide we're talking about. So if we're talking about DNA, DNA is our main nucleic acid. It's the one that's in the nucleus of our cells. It's the one that is going to store information about how to make proteins for the entire body in every single cell. DNA is deoxyribonucleic acid. So deoxyribonucleic acid, or DNA, so DNA. So our DNA, in this case, rather than having ribose, it's actually using deoxyribose, which is where the deoxyribo part of the name comes in. So the sugar is our deoxyribose sugar, which is just a different form of the ribose sugar. So our deoxyribose sugar. And then we have the same phosphate group that we already talked about. And then we have different bases. So our different nitrogenous bases. And that, again, is what makes up the nucleotide itself. So for DNA, we have adenine. That is one nucleotide base. We have guanine. That is another nucleotide base. We have cytosine. And then lastly, we have thymine. So if you have heard anything about DNA and you've heard the different letters, perhaps, this is where we get those. The adenine is the A, guanine is the G, C, cytosine, T, thymine. So these are our different nitrogenous bases. And so this is where we get our names for our nucleotides from. Now, if we kind of scroll up here a bit, I still want that illustration to be here. So we'll go up just a little bit here. If we take a look at the actual structure of DNA, again, we have our monomer to polymer structure. Our monomer is our nucleotide with our deoxyribose sugar, the phosphate, and one of our nitrogenous bases here. And what we see is that we have these, and they are going to form bonds. We are actually going to form a bond between the phosphate group of one nucleotide, and it's going to form a bond with the deoxyribose of another nucleotide. And that's going to form kind of the backbone of our DNA. So I'm going to kind of draw out, let's say this P is actually supposed to represent this phosphate group up here. So we have our phosphate group, and then we have our, let's say our ribose group there, our deoxyribose, okay. Then we have another phosphate group. Then we have our ribose, ribose. Then we have a phosphate group. So you can see that the phosphate group of one is going to be attached to the ribose or the deoxyribose of another. And we can keep going. So phosphate, and then keep going that way, and then we can keep going up this way as well to make our backbone of the group. What we're missing then here is our base, and our base is going to come off of here. Of course, we have the oxygen in here. And our bases are going to be the A, the G, the C, or the T. So we can just do one of each. So this will be our C for, let's say that's a cytosine nitrogenous group that's off of there. We can do the T here for our thymine group, and we'll do an A down here for adenine. So this is one side of what we normally think of as kind of a ladder. So if we think of DNA, if we think of it not twisted up, perhaps you've seen DNA before, it's similar to a ladder. So we have the side of the ladder where our hands would go, and then we have the rungs of the ladder. So the side of the ladder right here where our hands would go if we were walking up is actually what you see here. This is formed by the phosphate groups and the ribose groups. So if I just kind of turn these into circles, so our phosphate group and our deoxyribose, phosphate group, deoxyribose. So this right here is actually that right there where we're going to be holding on to. Okay, so we have one side. Let's pretend that it's just the side on the right for now. And then what's making up the rungs is actually split down the middle here. And right here, these rungs would be things like the A, the T, the C, and the G, our nitrogenous bases. So these would be individual nitrogenous bases here. And so DNA is actually composed of two of these strands. So we have one strand on this side here on the right, and then we have one strand on the left. So this is our phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And then here is our nitrogenous base, nitrogenous base, nitrogenous base, making up the rungs. And then on the other side, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, and then nitrogenous base, nitrogenous base, nitrogenous base. So we have our part where we would hold on with our hands. It's called our phosphate deoxyribose backbone. It forms the backbone of our DNA structure. And then we have the rungs in the inside, which are nitrogenous bases. So I'm going to scroll this up a bit here. All right, so we have our backbone, phosphates and sugars, and then we have our nitrogenous bases. So the important thing to understand then is that these two pieces, the left side and the right side, are two different strands. What's now holding them together in the middle here are hydrogen bonds between those bases, between the nitrogenous bases, which are the rungs in here. So we have our sugar phosphate backbone. We'll just simplify it here, right? Sugar, phosphate, phosphate, sugar. Okay, and then we have our nitrogenous bases sticking out. Our nitrogenous bases, again, remember we have an A, T, C, and G. What we see then is that we have our hydrogen bonds to the other rung, our sugars and phosphates here, over here. And they always are going to bond with the same letters, the same nitrogenous bases. What that means is that our G at the bottom here is always going to hydrogen bond with the C. So it will always be a G and a C. And then here, of course, our C is going to bond with a G. It will always be a C and a G. Here, with our thymine, it's always going to bond with an A. And up here with our adenine is always going to bond with the T, the thymine. So we always see A and T across from each other and C and G across from each other. Our cytosine, guanine, adenine, thymine. And that is because our adenine thymine actually has two spots where we can have hydrogen bonding. And so there are two hydrogen bonds between adenine and thymine, so they match up. And then there are three hydrogen bonds between our cytosine and guanine. And that is how they match up. So if we had a guanine across from an adenine, for example, they wouldn't match up because they have a different number of spaces for hydrogen bonding. So it would not match up, and we wouldn't see this ladder formation. Then, of course, we take our ladder and it twists because of the forming of these bonds. Okay, so the forming of these hydrogen bonds in here, and then the bonds between the phosphates and the sugars and the nitrogenous bases gives us that double helix, as it's called. Helix meaning that spiral notebook type shape. Double helix shape. Double because there are two strands that are attached to each other, and then helix because it's this spiral shape. So that's the structure of our DNA. And we find DNA in the nucleus. It is what's going to hold all of the information for all of the proteins in an organism. And then what we'll see is that we have our DNA. If we have our cell, it has a nucleus, and we'll talk about this in the cell section. Inside here, we have all of our DNA. Our DNA stays in the nucleus where it's nice and safe. It's protected. It's got a membrane out here. It's got another membrane on the outside of the cell. It's protected by all the stuff that's inside of the cell. It stays in there. But what we need to do is get the information from that DNA out so that we can make proteins with it. The way that we do that is we use RNA. Basically, we take DNA, we make a photocopy of it. That photocopy, that copy is RNA. The RNA can then go out here, where it then is used as a template for a protein. And we'll talk about that more when we get into cells as well. So generally, just know that DNA, we're going to make a copy of it, similar, but not exactly the same. DNA, make a copy of it in RNA, and then that RNA will go out into the cytoplasm, out in the rest of the cell, to make a protein. So the difference is then, there's very minimal differences between DNA and RNA. RNA then stands for ribonucleic acid. Notice we're missing the deoxy part that was here for DNA. And that's because instead of using the deoxyribose, of course, now we just have the plain ribose sugar. So our sugar here, remember that was a ribose sugar instead of deoxyribose. So one difference between DNA and RNA is that we have a ribose sugar rather than a deoxyribose sugar. So we have our ribose sugar. We still have our phosphate group. That stays the same. And then we have our nitrogenous base. That is the same. So this whole nucleotide structure, let me write that down, nucleotide structure. The three parts, there's still the same three parts, the phosphate group, the sugar, and the base. The phosphate is exactly the same between DNA and RNA. The sugar is different. It's a ribose instead of deoxyribose. And then the bases, we have a difference there as well. For our RNA, we still have our cytosine. We have our cytosine. Let me just erase that. That was a bad job there. And guanine. But we see a difference when we get to our adenine and thymine. Okay, so we said that we had A and T matched up in DNA, remember? So A and T matched up in DNA. In RNA, we still have our adenine, but instead of matching up with the T, we don't have the T. The thymine is not in our RNA, it's only in DNA. Instead, we have uracil. So instead of A and T, we now have A and U matching up. And we still have the C and G matching up. Okay, so those are our differences between our DNA and our RNA. And in RNA, we end up having different types of RNA. So again, our RNA is made in the nucleus from DNA. So we have our DNA. We're going to use our DNA as a template to make RNA. Then the RNA is going to leave the nucleus and go out into the rest of the cell. And then we're going to make our protein using RNA. Now, there are 3 different types of RNA that you need to know. The first one is called messenger RNA, also called mRNA. So messenger RNA is going to contain the information about the amino acids in the protein. So this is going to code for the amino acids, AA, in the polypeptide. So this is the messenger. So this is the one that's carrying the information, just like a messenger would. We're taking DNA, we're going to make our little messenger guy that's going to go out there and it's going to say, hey, these are the amino acids we need in order to make this polypeptide to make the protein. The next type of RNA is called transfer RNA, or tRNA. Our transfer RNA is going to bind to specific amino acids and match them up to the mRNA. So it's going to transfer the amino acid to the polypeptide. So it's actually binding them by matching mRNA. And we'll look at that in more detail in a couple of chapters when we're looking at the cell. The third type of RNA is ribosomal RNA. So that's rRNA. So our ribosomal RNA is actually part of the ribosome, and the ribosome is an organelle that we're going to look at when we talk about the cells. So this is actually part of the ribosome, and it's going to assist in joining the amino acids. So part of the ribosome assists in joining the amino acids in the polypeptide chain. So those are our 3 types of RNA molecules. So again, our RNA is made up of our nucleotides. Okay, so our nucleotides are the individual monomers, and then the polymer is the RNA. So our monomer is the nucleotide. And remember, the nucleotides are different based on whether it is an A, a U, a C, or a G, because we're talking about RNA, so we have the U. And then the polymer is going to be either mRNA, tRNA, or rRNA. Versus when we were looking at DNA, the monomer was the nucleotide, but the nucleotides include A, T, C, and G, and our polymer was DNA, just one, DNA. All right, so the last type of nucleic acid that we are going to take a look at is actually called ATP. This stands for adenosine triphosphate. ATP. So ATP includes a lot of the things that we've already talked about. So it has a base, but the base here is specific. It's adenosine. Okay, so its base is adenosine. I'll draw it over here just because that's where the other ones were. Adenosine. And then we have our sugar. The sugar in this case is the ribose. And then instead of having a single phosphate over here, this is triphosphate. So we actually have 3 phosphate groups here. So adenosine triphosphate. So this is another nucleic acid. It has a nitrogen-containing base, adenosine, the sugar, ribose, and then we actually have 3 phosphates. So it makes it different than our nucleotides in that it has these extra phosphates here. The function of adenosine triphosphate, or ATP, is to provide energy. So remember, our DNA and our RNA are for information storage. Our DNA stores the information. Our RNA is going to carry that information to our ribosomes to make proteins, or is the ribosome RNA our RNA, or is the transfer our RNA? The function for ATP, on the other hand, is to provide energy. This is our energy molecule in the cell. And so any process that requires energy is going to be utilizing ATP in the cell. So we need it in all of our cells. So ATP then is our high energy molecule. The way this works is that these phosphate groups and their bonds here is what is going to give ATP its energy. Okay, we actually see a reaction. I'm going to kind of scroll down here for a moment and draw something out. So I'm going to start with our ATP molecule. I'm going to draw it very simply though. So ribose, this is our adenosine, and then over here is a phosphate group, 1, 2, 3. So here's our ATP molecule. ATP, adenosine here, triphosphate, 3 phosphates. So our ATP molecule, if you remember what our phosphate groups look like, I'll just draw it over here. The phosphate group was a phosphate attached to a whole lot of oxygens. So roughly like this, because this oxygen is going to attach to the ribose, this one will attach to the next phosphate, and so then we have a negative charge here. If we have 3 of these, if you can imagine another phosphate just like this being right next to it, what we then see is that we have a negative charge, lots of negativity here with these electronegative oxygens right next to each other. They're forced next to each other. And this is why adenosine triphosphate, ATP, is a high energy molecule. What we see is that right here is where we have our energy stored. When we make the molecule ATP, what we do is we are adding these phosphates to the molecule. We're creating this bond between the phosphate groups. When we do that in a cell, when we add a phosphate right next to another phosphate, it's like taking 2 magnets and having them on the same side. So you have one on the positive side and the other on the positive side, and you're trying to force them together. If you've ever done that, it's very, very difficult. You're trying to push them together, and all they're doing is repelling each other, and you can't really push the magnets together. What we do in our cells is we do that with these phosphate groups. These phosphate groups are very negatively charged. So we have a negative side of a magnet, for example, and a negative side, and we're forcing them as close as we can, and then we attach them to each other in this phosphate bond, or in this bond between the phosphate groups. When we do that, it doesn't allow them to repel each other. If they're not able to repel each other, they've got this bond here. What that means is, another analogy, is that it's like a spring. If we take a really long spring, and say we take a spring that's really, 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 really long, and we push it with our hands, a very rudimentary hand here. We push it with our hands. this direction. We're trying to squeeze it as tight as we can, and we get it this tight, really, really, really tight, with our hands holding it together. And that's what we have, again, with the phosphate groups, is we've pushed this together. Now, if we remove these hands, right, if we take those hands away, what we have is a spring that's just ready to explode, right, and it's just going to burst out. And that's exactly what we have with these phosphate groups. We're forcing these phosphate groups kind of like a tiny spring in here. And so they're just holding all this energy. There's all this energy in there, and if we just do one little thing to release that, it's just going to burst out and release a lot of energy into the system. And so that's exactly what we've done with our ATP molecule. And so I'm going to erase my spring here, and I'm going to show you the reaction here. So what we have is our ATP molecule, and it can release energy. So what we do is we cleave off this phosphate group. We're going to go in and we're going to chop this part off. And what that does is it releases a whole bunch of energy. When we release that energy, then what we're left with is we still have our ribose. That's hanging out just the same. We still have our adenosine. We have our phosphate group here, and then we have our other phosphate group, but we have cleaved off this third phosphate group. So this phosphate group is hanging out over here, and it's negative. So at this point, then we have what's called adenosine diphosphate. The D in there, diphosphate meaning 2. So now we have 2 phosphates instead of tri for 3. And then in our cells, so if we need energy, we're going to take ATP and we're going to break off that third phosphate group, and it releases a whole bunch of energy in the cell for us to do other stuff, like build up other molecules. We need to put energy into those systems to build up other molecules. It takes energy to lift those building blocks. When we do that, then we're left with ADP now, which is a lower energy molecule. Now we just have this one spring in here instead of the 2 springs, so there's not as much spring energy bound up in there. And then we have the phosphate group, which has been removed. Then we can go in and through another process, we can put energy into the system. That's what we do when we eat food, for example. We take glucose and we take the energy that's stored in a glucose molecule, and we take that energy and we use it to attach this third phosphate again. So this part requires energy. So this is a release of energy over here. When we remove that third phosphate group and we get ADP, then through a process like breaking down glucose, we're going to take the energy that we get from glucose in order to force this third phosphate back onto the molecule, just cramp it in there, screw it on, and then make sure that it stays in place so that then this ATP can float around in the body and go to wherever we need energy. So this is our process, and this is what you need to know for ATP. That ATP is our high energy molecule. It's storing energy in these phosphate bonds here, and it's, again, like a spring. We're just crimping those. We're just holding them super tight, and then at any moment, if we release something, right, if we break that bond, we remove one of our hands, then it's going to burst out that energy. When that happens, that phosphate is broken off. We have our lower energy molecule, ADP, and then in the body, we're going to take energy from other sources. So for example, from our carbohydrates like glucose or from our lipids like fats. And we're going to use that energy to reattach the phosphate group. We're going to try to snap it back on there so that then we have an ATP molecule again that has a whole lot of energy that can then go around the body and can be used to release energy again for wherever we need it, whatever reaction needs energy. So ATP is our energy storage molecule. It's allowed to go around the body to wherever we need energy.